Now, fatty acids, they make the membrane less fluid than the unsaturated fatty acids. The fatty acids, they make them more fluid. And you'll get these kinks that will appear between the double bonds, keeping them from packing too tightly. Now, in animal cells, cholesterol must make up as much as 50% of membrane lipids in the outer outlets or leaflets. The cholesterol can fill up gaps left by unsaturated fatty acids. This has an effect by decreasing the membrane fluidity, but it increases the strength of the membrane. Overall, this is going to lead to a membrane with intermediate fluidity that is more durable and also less permeable. Now, when it comes to the different types of environments that a single-celled organism will encounter, if the temperature is, if we have an increase in temperature, meaning that it's hot outside or warm, we'll have an increase in fluid. Oops, that's an ID. But if the temperature drops, decrease in temperature, we'll have a decrease in the fluid. Now there are 6 functional classes of the membrane protein. So I have 6 main functions. So I'm going to we'll go through 2 of them at a time. We have transporters, we have enzymes, we have cell surface receptors, cell surface identity markers, cell-to-cell adhesion proteins, and the 6th one will be cytoskeleton anchors. So the first one, which is our transporters, these membranes are very selective. They're only going to allow certain solutes to enter or to even leave the cell. And they're going to leave the cell through channels or through carriers that are composed on these proteins. When it comes to the second one, our enzymes, now cells are going to carry out many of the chemical reactions on the interior surface of the plasma membrane using enzymes that are attached to the membrane. Cell surface receptors. Now on the surface of of the membrane, you're going to have these receptors that are going to sense these chemical messages. And they're going to detect by the receptor proteins that are anchored on their surface. The cell surface identity markers. These markers are going to identify, they're there to identify them to other cells. And now most cell types carry their own ID badge, their own markers. And the combination of the cell surface proteins and the glycoproteins that are characteristics of the particular cell type. So basically, if there are certain ions or solutes that are not to enter the cell, these receptors, these identity markers are going to, the receptors are going to say, hey, guess what? You're not allowed in here. So whatever, if there's other cells floating around that are allowed, or not cells, sorry, if there are other solutes that are supposed to come in, they'll let them in. The identity markers basically, hey, these cells are with us. We are the same type of cells that have this ID badge saying, okay, we are red blood cells or we are immune system cells. They're going to stick together and allow the communication between each other to occur. Then we have the cell-to-cell adhesion proteins. They use these specific proteins to keep themselves together. They're going to glue themselves together. Some are going to adhere by forming temporary interactions, where others have a permanent bond. And the last one is our cytoskeleton anchors. And these are surface proteins that will interact with other cells now to keep this anchoring pretty firm to the cytoskeleton. which is in the interior aspect of the cell, which will help with linking proteins. Now here is a diagram that your textbook has on page 100, figure 5.6, and it talks about the functions of the plasma membrane. So we already talked about our transporter, correct? We have solutes that will enter and leave the cell, and here's one of the channels that would help with transporting the solute from outside the cell to inside of the cell. And here's our enzyme, and they're the ones that are going to carry the chemical reactions to the inside of the cell, and as well outside. Oh, sorry, inside, sorry, and then outside. All right, then here's our cell surface receptor. They're the ones that are going to detect what needs to enter the cell or what needs to leave it. Okay, so if a calcium is here and it needs to enter the cell, and this is a calcium channel, then the calcium will enter and be able to pass through. But if it's a sodium channel, if it's a sodium ion, and this is still the calcium channel, he will not get through. Okay, and here we have our cell surface identity markers, which are going to identify with other cells. We have our cell-to-cell adhesion. I am going over it again to reiterate it, which are going to use certain proteins to glue themselves together, which is great. So we have two cells binding, and either going to have a temporary reaction or a permanent bond. Then we'll have, sorry for all these pop-ups, then we'll have our cytoskeleton anchors. And here they are right here. And this is just to make sure that the cell is anchored firmly to the inside of the interior aspect of the cell. Alright, so structure relates to the function. Now, there are a number of structures that the cell has within the membrane, and their role is very important. Now, they do have a diverse function, and one of them would be the peripheral proteins. They're the ones that are going to anchor the molecules that are attached. That they anchor the molecules attaching membrane proteins to the surface. Then we look at the anchoring molecules. They're modified lipids, and they would have a nonpolar region that will insert into the internal portion of the lipid bilayer. And then the chemical bonding demands that link directly to the proteins. Back. Then we're going to look at our transmembrane. Now, cell membranes that contain a variety of different transmembrane proteins, which differ in the way they traverse the bilipid layer. Now, the primary difference lays in the number of times the protein crosses that membrane. Now, each membrane spanning region is called the transmembrane domain. Now, these domains are composed of hydrophobic amino acids, usually arranged in alpha helixes. Now, the proteins need only a single transmembrane domain, to be anchored in the membrane, but they often have more than one such domain. An example of a protein with a single transmembrane domain is in the lincoln protein that attaches the network of the cytoskeleton to the interior of the plasma membrane. So here we have this spectrum, and we see here's our interior, and here is our exterior, and you see that they will sit here and start to form a little network to help with attaching to the interior aspect of the membrane. The membrane also has pores. When we talked about the nuclear pores, now we're talking about pores that are on the membrane. Now, some transmembrane proteins, they have extensive non-polar regions with secondary configurations of beta-pleated sheets, which are instead of the alpha helixes. Now, the beta sheets, they form a characteristic motif, folding back and forth in a cylinder, so the sheets arrange themselves like a pipe, which we see here. And they're going to arrange this pipe running through the membrane with the polar environment in the interior of the beta sheets spanning the membrane. Now this is called a beta barrel, and it's going to open up on both ends. Now this is a very common feature of a porin class of proteins that you will find within the outer membranes of some bacteria. And these openings, they're going to allow water molecules to pass through the membrane.